Okay, welcome everyone. We are in the week 4 of this course. I hope I am audible and my slides are visible to all of you. So, week 4 talks about the protein isolation and characterization. So far, we have covered till week 3. In week 1, we discussed about the characteristics of amino acids and a fundamental of kind of proteins. Then in second week and third week was more of the fundamentals related to the protein, their structure, 3D geometry, protein chemistry, and other characteristic about the protein. So now we are moving ahead, and now we are in the case to understand, to purify the protein, to isolate from the organism, and characterize it, whether which protein it is or how to what are properties of this the protein of interest that you are going to study. So we will talk about these all aspects in week 4. I hope you all have gone through the lectures that have been uploaded already in the NPTEL. So I will be now going directly to discuss about the concept that have been discussed in week 4. Before that, I would like to welcome all of you. I am Karan Kumar, Prime Minister's Research Fellow at IIT Guwahati, and today we will discuss about the week 4. So in week 4, the concepts were covered related to the protein isolation, and that includes the source of organism from where the proteins can be isolated. This can range from the microorganism to organism. Then the general strategy behind the protein isolation. Then moving forward, we discussed protein purification. This purification steps include salt fractionation, centrifugation, diffusion, sedimentation, chromatography, dialysis, and various type of other techniques. Then biophysical methods we also studied after uh, like when you have purified the protein, then how you will characterize it. So biophysical methods are mostly related to the characterization of protein, which include the different spectroscopic and spectrometric technique, then electrophoresis, isoelectric focusing, microscopic methods, and many more. Few of them you studied in the previous lectures also, previous module. That includes the X-ray crystallography and NMR solution to study the 3D structure of protein. So the important keywords that are that are there in the week 4 is protein purification, salting in, salting out, centrifugation, sedimentation, fluorophore, quenching, PRET, which is fluorescence resonance energy transfer, then quantum yield, Sds Space, isoelectric point, microscopy, tapping probe, FTIR, circularly polarized light, deconvolution, and many more. So I hope you all have gone through the lectures that are already uploaded in the NPTEL portal, and I hope you have understood it. So today we will be discussing the assignment problems that will not be entirely on the current assignment, but the this course was run. This course was already run in the previous sessions. So I will be discussing the assignment problem of week 4 from the previous sessions. So the contents of PPT. This PPT content practice questions on current module, then discussion on important topics, and also if you have any doubts, I will take into that. I will discuss those, and if you have gotten into some like you have not understood some concept, you can also ask question at the end of this presentation after we finished with the practice question. Then I will also discuss few of the important important scope of this module with research perspective, and also give you the research problems and its example that I am working on. So your first question for this module is, in isoelectric focusing, proteins are separated on the basis of their. Your options are relative content of positively charged residues only. Option B, relative content of negatively charged residue only. Option C, size. Option D, relative content of positively and negatively charged residues. So can somebody unmute themselves and try to answer this question? Anyone? Anyone can try? Yes. Sagar or Uday Bhanu? What could be the possible answer for this question? In isoelectric focusing, proteins are separated on the basis of options are relative content of positively charged residue only. Option B, relative content of negatively charged residue only. Option C, size. Option D, relative content of positively and negatively charged residue. Anyone of you can attempt this question? Okay, so let me answer this. The correct answer for this question is option D. Option D, which talks about the relative content of positive and negatively charged residue. Okay, correct answer is option D. What do you understand by isoelectric focusing? Isoelectric focusing is kind of 2D gel electrophoresis in which first the proteins are separated based on their charge. So this charge is imparted due to the different amino acid residue present in them, because we have studied the amino acid residue have different various amino acid residue have the different characteristics, and they can have the positive or negative charge based on their side chain. Okay, so in isoelectric focusing we use a ampholyte strip where we will put our solution, and then we will once the protein is aligned based on their charge group according to the pH gradient. Like the first is this is the pH strip. In this pH strip, what happens? The pH strip why this pH strip is created? Because the different charge group is aligned on this pH strip, and then when the protein solution is passed the protein solution is passed through this strip, based on their pi value they will accumulate according to their pH range, and then you will use this strip on the Sds Space, general Sds Space electrophoresis unit, where the first separation is based on the charge and then their separation is based on the molecular mass. Okay, this will be the high mass, and then the bottom side there will be low mass. So your correct answer is option D. Second question in this series is the fluorescence. The rate of fluorescence resonance energy transfer depends upon. So your options are, and there are three statements given, and based on that the options are designed. Your first statement is the quantum yield of the donor. Your second statement is the distance between the donor and accepted molecules. And option 3 is the relative orientation of donor and accepted transition dipoles. So who is willing to answer this question? Please unmute yourself and answer this. Anyone willing to answer this question? Please unmute yourself and answer. This involves the concept of fluorescence resonance energy transfer, in short, FRET.
Sir, option C.
Option C. Okay. Option C, which talks about the quantum yield of the donor, distance between the donor and acceptor molecules, and relative orientation of donor and acceptor transition. Can you please explain this question? Explain this answer. Why do you think so? What happens in FRET? Yes. Please go ahead. Your answer is correct. Yeah, but please give a rationale behind the answer. Why do you think all three are important for FRET? Okay, let me discuss the FRET concept of FRET. In FRET, this happens not because of the transfer of electron, but transfer of energy. In this, one donor molecule is there and one acceptor molecule is there. Both this FRET will only happen when the Foster distance, when their distance between these two molecules are less than the Foster distance, which is R0. R. Okay. R0 is the R0, capital R0 is the distance between the two molecules when the energy transfer is half of its maximum, which is 50% energy transfer. Okay, so the second option is correct, right? This one, second statement is correct. Distance, this particularly depend on the donor and acceptor molecules. Now, let me check about the relative orientation of the donor and acceptor transition dipoles. So this is also necessary because what happens when you put a molecule, when you have two protein, and in that protein you tag two fluorescence molecule which have the significance spectral overlap in their emission spectra. Means the first one will be the donor. In donor, the emission spectra of donor should overlap with the absorption spectra of the second fluorophore. Okay, so in this, what happens when there is a spectral overlap, means the emission spectra of first will overlap the second spectra, then you will see the FRET. Okay, and if both, let us see, let us assume the first donor, the first fluorophore is this one, which is donor, and this is acceptor. So if donor is on the left hand side, then the incident light will fall on it, then it will emit a light, which is emission light. This emission light should excite the acceptor one, which is second fluorophore, then only it will emit the light. So that is why the orientation is also very much important. Then the quantum yield of the donor, the intensity of this energy released should have the appropriate quantum yield to excite the acceptor fluorophore. That is why the statement 1 is also correct. So rate of FRET depends on all three conditions. So that is why the option C is correct. Your next question is, in an experiment, at which wavelength of the absorbance is measured on the sample protein BSA? Can you please answer this question?
280.
Anyone? 280 nanometer. Okay. Why?
Because purity of protein is detected, I think, at 280.
UV. Why? At which amino acid or which backbone absorbs at 280 nanometer?
Aromatic amino acid, 280.
Aromatic amino acids. Can you please name some of them?
Tryptophan.
And?
Phenylalanine.
Yes.
Tyrosine.
And? Good. Very good. All three aromatic amino acids absorb maximum at 280 nanometer. This 280 nanometer is majorly related to the tryptophan absorbance. Others absorb more or less than, but if you consider the average of all three aromatic amino acid, then the maximum absorbance happens at 280 nanometer. That is why we will take the absorbance at 280 nanometer. Your answer was correct. So now your next question is, in Sds Space, protein sample is first treated with detergent sodium rhodosyl sulfate in order to. Your options are to render the protein with negative charge, or to render the protein with a positive charge, to renature the protein, or adjust the pH of protein. What is the correct answer? Option A. Option A.
The
Why?
protein with a negative charge.
Can you explain the concept of Sds Space? What happens in Sds Space?
Actually, protein, I think, having variety of amino acid with the variety of charges. So
Okay.
I think during SDS page, protein transported or migrated via gel on the basis of molecular weight, and when we pass the electric current from one electrode to another, so migration may happen, uh, I think, toward the positive electrode
Okay.
when there is a whole complete negative charge over it.
Okay. Good. So what is the property of Sds Space?
SDS page can denature the proteins, I think.
Okay. And it is a
And
negatively charged detergent. The main
negatively
important, yeah. So that is why
yeah.
it denatures this protein and then it accumulates around the protein, and
Yes.
that is why it makes a globule kind of thing. And all the proteins, all the molecules that are under the analysis, they will exert a negative charge. So that is why the correct option is A. Okay. Your next question is gel filtration chromatography separates protein based on their which property? Is it isoelectric point (pi) or secondary structure content or effective size or morphology? What is the correct option according to you?
I think effective size.
Effective size. Why do you think so? Why not secondary structure, morphology, or PI?
Uh, Gel filtration chromatography having beads, those with a particular means gel having particular pore size. So definitely pores within the gel affects on the migration. Uh,
Yes.
so there is no issue of PI because PI depend upon charges.
Yes.
Secondary structure content is a different fact because we cannot think about any structural information in filtration or chromatography in this
Yes.
case.
Accurate.
And morphology, I think this is a very broad issue.
Good. So your answer is correct and explanation is also correct. So the correct answer is effective size. Very good. Now the next question is this is a true false statement question. Your statement is in a protein when tryptophan residue becomes hydrogen bonded or is exposed to water. That means the tryptophan is on the outer, like when the protein unfolds, then water, when the tryptophan comes in contact with water, the emission shift to longer wavelength. Is it correct or false? Is it true or false? What happens when the protein opens up and tryptophan residue comes in contact with water and forms a hydrogen bond? What is the main characteristic of emission during that time? Will it shift to longer wavelength? True or
I
false?
think false. I think false. It is not true. I
Okay,
think
why?
because most of the time we can use water as a solvent, or here
Okay,
there is no such kind of interaction possible. Those can be shift the wavelength, absorption wavelength.
okay. Unfortunately, your answer is wrong.
So
This is a
okay,
true statement. No problem.
okay, yeah,
What happens when the tryptophan residue is exposed to the water solution? It means the hydrophobic, then the hydrophobic interactions are there, due to which the residue, this tryptophan residue, the excitations also get shifted to right side. Okay, so it will shift to the longer wavelength, and this statement is correct. Okay. Now your next question is in anion exchange chromatography. Please note this is specifically to the anion exchange. Okay. And this is a category of ion exchange chromatography in which we are talking about the anion exchange chromatography, right? So in anion exchange chromatography, your options are, which is the correct statement? The column contains negatively charged leads with where positively charged residue, positively charged protein binds, or the column contains positively charged beads where the negatively charged protein binds, or column contains both positive and negative charged beads where the protein binds depending on their net charge, or option D, the column height is utmost important in the process. There is a brief diagram is also given of the anion exchange chromatography. Please try to answer this question. What could be the possible fate in anion exchange chromatography?
I think A. The column contains
A.
negatively charged beads where positively charged protein can bind.
Okay. This is here you can see, right? It is already given here in the figure. The beads are mostly positively charged, right?
Yes,
And the
yes.
mixture of protein will be negatively charged. So your correct option is B. In anion
Okay.
exchange, you can remember this by interchanging. Like anion exchange chromatography means the beads should be positively charged and it should be bounded with something. But in what happens when the higher negatively charged protein comes, then it will replace the bounded proteins that is bounded on the charged bead, and that is why this anion exchange, that is why it is called the exchange, means there will be exchange of more anionic protein, means negative charge. So that is why the option B is correct. The column contains positively charged beads and negatively charged proteins binds to it. Okay. Your next question is related to the CD spectra. So in the CD spectra of protein, identify spectrum marked here. So identify X, Y, and Z. What does it denote? Is it X beta sheet, Y alpha helix, Z is random coil, X is alpha helix, Y is beta sheet, Z is random coil, X is beta sheet, Y is random coil, Z is alpha helix, X is random coil, Y is alpha helix, Z is beta sheet. Uh, can somebody attempt this question? Anyone? Okay. So your correct answer is B. X represents here alpha helix because alpha helix have the two in CD spectra. You will see alpha helix gives two peaks. Okay, at around one around the 220 nanometer, another around 205 nanometer, 205 or in the range of 220, 210 nanometers of wavelength. However, this red represents the beta helix. It has only one peak. Okay. This is, as you all know, the CD spectra is related to the absorbance of like the change of the absorbance of the circularly polarized light, and it is the difference of the left-handed circularly polarized light and right-handed circularly polarized light. Okay, so your correct option is B, and random coil is represented by Z. Okay. Now, your next question is the simplest way to remove ammonium sulfate from the system is. You have given a sample in which you have contamination of ammonium sulfate and you want to remove ammonium sulfate from the solution. How will you do that? Will you apply precipitation or dialysis or chromatographic technique or purity checking? What will you do?
Dialysis.
Dialysis, okay. Can you give a brief about the dialysis? What happens in
Uh,
dialysis?
I think dialysis membrane having different pore size as per their molecular size weight, and
Okay,
after selection of proper pore sized membrane, we can uh, separate the salt from protein, which is
okay,
precipitated in the solution
okay. And can you
within
please
24
specify?
hours.
Okay, can
Yeah,
you please specify which kind of membrane do you use in dialysis? Which
it's
kind of membrane?
I think like polyethylene membrane are there.
No, no, no. I am talking about the main property of the membrane. What
Membrane.
is the property of membrane? used in dialysis?
I have used, but I do not read about it.
Okay, no
Yeah.
problem. So the membrane that is being used in dialysis are semi-permeable membrane. Those are termed as the semi-permeable membrane. They selectively transfer, they selectively allow the passage of molecule, and dialysis is used for the uh, separation of low molecular weight or lower size compound. Ammonium sulfate is a very low, like it is the size of ammonium sulfate is very low. So based on the pore size of the semi-permeable membrane, we can remove the ammonium sulfate from the system. So correct answer is B, which is dialysis. Now the next question is in the first step of two-dimensional gel electrophoresis. So as the term represents the two-dimensional gel electrophoresis, which means it will involve the isoelectric focusing and Sds space gel electrophoresis, right? So it generates the series of protein bands by isoelectric focusing. In the second step, the strip of the gel is turned 90 degree and placed on the another gel containing Sds. Okay. The electric current is again applied in the second step. That means it is talking about the SDS page electrophoresis, right? So what would be the fate of the sample? So the protein with similar isoelectric points become further separated according to their molecular weight, or the individual bands become visualized by the interacting with protein specific antibody in the second gel. The individual bands become stained so that the isoelectric focus pattern can be visualized, or the proteins in the band separate more completely because of the second electric current is in the opposite polarity of the first current. So what is the correct answer for this question according to you?
I think A. Proteins with
Option A.
similar isoelectric points become further separated according to their molecular weight.
Okay, very good. This is the correct answer. So, and explanation is already in the question. So, and we have also discussed this in the previous questions. Now, moving to the next question. Which among the following is incorrect about the gel filtration chromatography? Okay, so your options are particle size determines the void volume V naught, or Vt, which is the total volume of the column, minus V naught, which is the particle size, is the volume occupied by the gel space including matrix, or the partition coefficient KAV is inversely proportional to the log of molecular weight, or option D, the partition coefficient KAV is equal to VE minus V naught divided by VT minus V naught, where V is the intermediate volume. So what is the correct statement? Oh, what is the incorrect about the gel filtration? This is talking about the incorrect. So what could be the incorrect statement in this question? Have you remember, are you have you remember the gel filtration chromatography? What happens in gel filtration chromatography? We have just discussed in some previous question.
I think particle size determine the void volumes is incorrect. Maybe.
Particle size determine the void volume V naught is incorrect. Why do you think so? The incorrect statement here is option C, which is the partition coefficient Kv is inversely proportional to the log of molecular weight. As we all know, in the column, when you put the beads into it, then all the beads are then the complete volume is represented by the Vt, okay, which is the column size. The option B, like option B is correct. The total volume is Vt, then V naught. V naught is nothing but the particle size particle size. Okay, V naught means the size of this particle, and V is the intermediate volume. Okay, so and the partition coefficient is given for this column. Partition coefficient for this particular chromatography is V minus V naught divided by Vt by V naught. This is also a correct statement. So These three are correct. And if partition coefficient is the formula of partition coefficient is given by this, then obviously this statement will be incorrect. So correct. The correct answer for this question is option C. Right. Your next question is, which would be the best to separate the protein that binds strongly to its substrate? Okay, so you have given a sample in which a protein, you need to separate a protein that binds strongly with a substrate. So based on this characteristic that you know, which kind of chromatography you can use to purify this protein? Will you use the gel filtration or affinity chromatography or cation exchange chromatography or anion exchange chromatography? Please note, strongly bind to substrate. Where is my yeah? Strongly bind to its substrate. Anybody want to attempt this question?
Affinity chromatography.
Affinity chromatography. Accurate. So this is self-explanatory. Gel filtration is to filter the low, high, and medium molecular category. Then cation exchange. This question is talking about the protein that binds strongly. But however, the cation exchange and anion exchange involves the charge kind of like it involves the charge interaction. But when it is talking about the strong bind, that only happens in the affinity chromatography, right? So the correct option is B. Your next question is, the purity of an enzyme at various stages of purification is best measured by option A, total protein, option B, protein percent recovery of the protein, option C, specific activity of enzyme, option D, percent recovery of the enzyme. How you can measure the purity?
Specific activity of the enzyme.
Accurate. Very good. So moving to the next question. In a mixture of the 4 proteins listed below, which should be the elute second in the size exclusion gel filtration chromatography? So to answer this question, you need to remember the concept of size exclusion chromatography, right? Or gel filtration chromatography. Your options are cytochrome C, which have the molecular weight of 13,000 Dalton, means 13 kilodalton. Then immunoglobulin G, which have the molecular weight of 145 kilodalton. Then RNA polymerase having the molecular weight of 450 kilodalton. Then serum albumin having the molecular weight of 68.5 kilodalton. So which of this will elute second in the gel permeation chromatography, size exclusion chromatography? Anybody want to attempt this question? What is the principle of size exclusion chromatography? Which one will elute first?
I think large sized particles.
Yes. So so in this question, which one is the highest molecular weight?
RNA polymerase.
RNA polymerase, correct. So now in the question we are asking the second which one will elute second. So the correct answer is immunoglobulin G, right? Which yes, because it has the second most highest molecular weight among this option. So correct option is B, right? Question number 40. Now the question is given as, you know the protein you want to purify from a natural source which forms a multimer with multiple subunits giving a molecular weight in a solution much bigger than the visualized denatured on Sds Space. Now there is only a small amount of target protein in the total protein sample. Which of the following is an appropriate purification strategy? Means which one you will follow first and then you will follow second, or you will follow only one chromatography technique to purify the sample. Your options are, option number 1 is SEC, which is size exclusion chromatography. Option B, you will first use the ion exchange chromatography, then you will use size exclusion chromatography. Or option C, you will use only ion exchange chromatography. Or option D, you will use affinity chromatography followed by the size exclusion chromatography. So read this question carefully and try to answer this. Please remember the question is about the protein have the multimers and it has the multiple subunit. So if you use the Sds Space, it will denature the protein, right? Then you will need to there is only a small amount of target protein in the total protein sample. Which of the following is the appropriate purification strategy? What will you follow?
I think B. IEX
B.
followed by ISCC.
Okay. How this will help you?
We can separate the protein as per their charges, and then we
Okay.
go for the separation and purification by using SEC, I think.
Yes, your answer is correct actually. First, you will follow the IEX, means ion exchange chromatography, and then you will follow the site exclusion chromatography, which because it has the multimers. So if you follow the STS page, it will denature, but it has the solution which is much bigger to visualize. So obviously, you will go to the chromatography techniques. So in the chromatography technique, because this has the multimers, so when you will use the IEX, then the protein will separate based on their charges or isoelectric point, right? And then followed by the size exclusion, which means the bigger protein will elute first and then coming forward by the other proteins, right? So your correct option is B. Next question is the salting out process with regards to the proteins mainly involves precipitation of protein using the sodium chloride or precipitation of protein using copper sulfate or precipitation of protein using ammonium sulfate or none of these.
Precipitation of proteins using ammonium sulfate.
Correct. This is a fact-based question. So your your answer is correct. 15 is C. Now coming to the next question. Primary step in protein purification include differential certification, homogenization, solubilization, or extraction. What are the primary steps? So the options are 1, 3, 4. Options B is 1, 2. Option C is 1 and 4. Option D is 1, 2, 3, 4. What are the primary steps?
D. 1, 2, 3, 4.
Very good. All four are the primary steps in protein purification. Now your next question is, you have given a insulin hormone and you need to identify, you need to purify it and purified it with the receptor. So this will be an example of which kind of chromatography technique. Is it ion exchange or affinity or gel filtration or ligand mediated? What is the correct answer for this question?
Ligand-mediated chromatography.
Ligand mediated. Why do you think so?
Because if suppose receptors may bind ligand, I think. So insulin may consider as the ligand. So insulin when bind to the receptor, this complex, ligand receptor complex. So same
Okay.
property we can utilize in ligand mediated chromatography. Maybe.
Yes, your analysis is correct, but in this case, the receptor and insulin hormone both are the bigger molecules, right? So in this case,
Affinity.
you cannot treat the insulin in the as a ligand, right? Receptor is receptor, yeah, correct. But in both of them, because the insulin is specifically to its receptor, like its affinity is towards its receptor, so that is why ins this technique will be called as the affinity chromatography because it has the affinity towards it, right? So this chromatography will be the affinity chromatography.
Yes, yes.
So now we are done with the questions about it. Now we are moving. If you have any doubt related to the previous sessions, you can ask me. like or the doubts related to this session, or the concept you have not understood well, you can ask, and then we will go on to the to discussing about the research perspective of this module. Any doubts you have, anyone?
Sir, I think
Yes.
question answer session is best. But I have one doubt. My doubt related with the protein preparation for LCMS.
Okay.
So I
Okay.
think
LCMS protein preparation. Okay.
protein preparation. So when we take the tissue from the
Mm-hmm.
animal like fish, suppose, and after
Okay.
taking tissue, we have preserved it for few hours in liquid nitrogen, or we can carry it in dry ice.
Okay.
Then we bring it laboratory and we try to prepare the protein, and we
Mm-hmm.
having aim to study the global proteomics.
Okay.
So what are the strategies to prepare the sample for
Okay.
LCMS? Yes.
For LCMS is related to, yeah, when LCMS is a combination of HPLC and MS, right? Mass spectroscopy. HPLC and coupled with the mass spectroscopy. So in this, the separation, the identification you can carry forward. So in that, the HPLC will separate the protein in the different this is a chromatography, right? So based on their property, the absorbance or any HPLC the strategy, based on that they will first separate, and then the mass spectroscopic technique will be used for the analysis to identify what kind of band it is, what kind of protein this it has, okay, in the sample. So the preparation step of the sample preparation step will contain, first you need to homogenize the sample, then you first you homogenize, then extraction, and this whatever the primary step that you follow that, and then finally you do a simple chromatography. If you and try to make it the smaller in size, then filter it to remove all the unwanted material, like when you like using the centrifugation, when you homogenize it, then you use then you dissolve it, and then finally you make it the sample as liquid as possible, because in HPLC you usually in LCMS, whatever you are talking about, the LC stand for the HPLC, which is the liquid chromatography, right? So in HPLC, you need to have the samples which is well below the is well mixed. Okay, so you need to prepare your sample accordingly so that it only in a it exists in only the liquid phase. Then you inject the sample in the LC like HPLC, and then it will separate the band. So this will be the correct strategy for preparation of the sample. You must filter this with the 0.02 micron, 0.02 micron microfilter, syringe filter, so that this your sample should not contain any solid particle which can obstruct the column that is there in the HPLC, right? In HPLC you use a column, right? So which is the solid state, solid phase, stage, or also the stationary phase. Then you will connect this by a mobile phase, and in that mobile phase you will inject your sample. Then based on the affinity or whatever technique, whether it is ion exchange chromatography or the affinity chromatography, whatever the principle you use in the stationary phase, that will resolve your sample in the component that is there in your sample. Okay, so because you are passing your sample through the stationary phase, it should not contain any solid particle which can obstruct the flow of the liquid. Okay, so you will filter with 0.2 micron syringe filter, and then you pass the sample. The detailed preparation is related to your kind of protein that you are using for your sample preparation for LCMS, and that you can find in the respective study, like research paper you can target, and then from there you can prepare accordingly. The general step is like this only. What you prepare using primary step, and then you follow like a low resolution technique such as different chromatography technique to separate out the sample, and then you use the sample, you filter out the sample and then put into the LCMS. Okay, I hope I am able to clear your doubt.
Yes, sir. One more short question.
Yes, go
Uh,
ahead.
Targeted proteomics. I must to know about what is targeted proteomics and what is the difference between targeted and global proteomics?
In targeted proteomics, you need to know the basic principle between the basic difference between the targeted proteomics and global proteomics. In global proteomics, you have not specified which kind of protein you want to study. You will let us say you have a organism or microorganism, let us say bacteria, and you want to access during a kind of metabolism or any kind of activity, like if you want to study what are the protein involved in the stationary phase of this bacteria, during the stationary phase which all proteins are expressed. So in that case you will arrest, and if you want to profile all the proteins that are involved in the stationary phase, then you will go for the global proteomics. Or else if you want to if you know that you want to study only particular kind of proteins, and you want to see whether this with this protein is involved in the that particular state of the organism, then you will go for the targeted proteomics. Am I able to clear your doubt?
Yes, sir. One more question is there.
Okay.
Uh, Databases are available on the Pride or different databases. So
Okay.
I think those files we access for our research uh,
Mm-hmm.
may having data concerned with the many proteins, means near about for my work 17,000 to 20,000 work per sample.
Okay. Okay.
So it is always variable, maybe up to 30 to 40%.
Okay.
Uh, so initially we start with a broad level of miss big sample, and then finally we get thousands of protein in a single
Mm-hmm.
sample,
Mm-hmm.
which are those identified. And when we try to compare few proteins for their properties or desired goal, so what happen? Quantitative values are very much variable in final analysis.
Mm-hmm.
So how we can compare it with the standard uh, values, those are available with the research papers.
Okay, this is a very specific question to a particular kind of research. So, and to answer this, I would like to recommend that the protein that you are targeting or the database you are targeting, you first study you first see the database which kind of proteins are involved, and for your particular kind of study, if you are let us see, you are targeting a certain type of protein which falls into a particular category, then you only target those kind of database which is focused on your target study, like your target type of protein, and then you match with those kind of database, because there are different databases, right? There are not only one type of database, there are multiple databases for various category of protein. So based on your requirement, you you search your uh, protein of interest in the particular database. That will result you with less time. It will consume less time, and you can use the bioinformatic analysis to minimize your time. Okay.
Thank you, sir. Thank you.
Okay. So now next, coming to the research perspective of this question, I will, and this is mostly related to the deconvolution and molecular docking. So you can go through my research Google Scholar profile, and you follow this. The link is given here. you can follow this paper, which I am going to discuss in this lecture. This is related to the mechanistic investigation of sonoenzymatic synthesis of n-butyl. So here in this paper, what I had done, right? My target was to study whether this n-butyl is a energy molecule. Like this is a molecule which have the various different role in energy sources. So, and this levulinate is synthesized based on the esterification process. Okay, and here we use the lipase. So in this paper, in this work, what we have studied the esterification of levulinic acid with n-butanol in presence of a lipase, which is Candida antarctica lipase B CALB. Okay, so to support the proof or to verify the mechanism of esterification, we performed the molecular docking simulation. So in that molecular docking simulation, and also we used here the sonoenzymatic. Sonoenzymatic means ultrasound mediated process. So in ultrasound mediated process, what we observed that without the application of ultrasound, this was giving certain kind of yield of n-butyl levulinate, but with the application of ultrasound, the yield was increased. Okay, so to verify why this is happening, we must know the esterification esterification mechanism and how the ultrasound is affecting the synthesis of n-butyl levulinate, that we understood by analyzing the 3D structure of lipase, Candida antarctica lipase. So for this, we studied the structure of without treated, like untreated lipase and treated lipase. Treated lipase means with sonication or without sonication. Without sonication is untreated, which is black one. This is the FTIR spectra of that particular protein. So ma'am has taught us that decolumulation of FTIR spectra result can tell us which kind of what are the content of the secondary structure that are present in this particular enzyme or protein, right? So, and that ranges from this is result of the amide 1 stretching. Like amide 1, there are 3 type of vibrational spectroscopy in FTIR. Amide 1 band result in the, if you see amide 1 ranges from the 1610 wave number to 1619 wave number, centimeter inverse. Okay, this is not wavelength, this is wave number. So, and you record the absorbance. FTIR gives you transmittance, then you calculate the absorbance based on that, and then in the range of this, you deconvolute the spectra. So the different wave number ranges give you the different content of the secondary structure. Okay, so here in the amide 1 fitting, what we observe that in the control, this is the control. B represent the control without sonication, and C represents the control. C represents the with sonication. So what we observed in the sonication category, the alpha helix content increased from 23.1% to 38.1%. First observation. Second observation, beta sheet content was decreased from 22% to 14%, and random coil percentage was also increased. There was a concurrent increase in random coil. So what is the conclusion of this? The conclusion of this that when we observed the structure of lipase, which is Candida antarctica lipase B, this is the 3D structure of lipase. So when we studied the structure of this, then measure of the alpha helix content was increased. This alpha helix content was involved in the formation of lead. Lead means where the protein, because the protein have this lipase particularly have a lead kind of a structure, which allows the passage of levulinic acid and butanol to its active site. Active site is inside this pocket. This is the active site, and this lead is mediated by the alpha helix. So what we observe, when the ultrasound is there, then this lead, the alpha helix content is increased, and the lead opens up, and all the binding pockets, like the acyl binding pocket, which binds the where the levulinic acid binds, and alcohol binding pocket where the butanol binds, these all the binding, this amino acid residue lies in the random coil. So increasing random coil means this structure will be relaxed, and the increase in alpha helix content means the binding pocket will binding lead will open up. So based on that we observe, based on that we postulated that this could possibly enhance the activity of lipase, thereby the N-butyl livinate, the yield of N-butyl livinate getting increased. So this is how you can study. This is how you can study the deconvolution and structural perspective to study the different kind of reaction mechanism related to the protein chemistry. So here you can see this is the levulinic acid bound in the pocket of this CALB enzyme, and this is the 2D representation. Then here in C is the binding of butanol in the binding pocket of this particular structure. Then here this is the transition state. I have not given the full mechanism of this in this slide, but you can refer my paper. You can read this paper, Mechanistic Investigation in Enzymatic Synthesis of N-butyl livinate. Here I have described the complete feature of the binding and how you can reduce the mechanism. So this is how we can use the deconvolution technique to study the protein chemistry. So now this is my information. These are the references. This link you can follow to open the particular paper that I have discussed, and these are the other references that I have taken into the consideration. The important announcement I want to make is let me share my screen. Yeah, here. Please regularly follow the announcement page and do submit all the assignments that are given. Okay, into the stipulated time. You can check your score in the progress tab. And also, if you want to ask any question related to this lectures, you can ask in the forum. Okay, the forum is this one. The forum you can navigate by clicking ask a question. This will redirect to you the forum of this Google forum, and there you can paste your question. The TA involves in this, like TA including me, will answer your question, and regularly I post, I send the message related to the YouTube link, whatever discussion we are making through. I will upload this into my YouTube channel, and then the slides also I regularly share. And this is the Telegram channel you can join for the elaborated discussion. If you have elaborated questions, you can and related to the previous tutorial that we discussed, there I asked whether you want to have a separate section on molecular docking and cooperative protein modeling computational biology technique. So I can take the session apart from the regular sessions if the time permits, and if many of you are interested. For that, you need to comment your question. Comment your question in the comment your agreement to this YouTube video of the week 3 that I have uploaded into my channel. Please subscribe to this channel, NPTEL-PMRF TA NOC22-BT62, and comment if you want to have a separate lecture on molecular docking simulation or protein biochemistry like computational biology. So, and regularly check this session, join the session on the same, using the same link. Okay, if you so now we are done with the session. If you have any question, you can unmute yourself and ask related to this problem solving session or related to the lectures, or you can also send me an email on karan18@iitg.ac.in. So is there any doubt?
Thank you, sir. So I think a separate session related with the molecular docking is essential for us.
Okay, so for that, you please comment on the video, and also if there are more number of like people are asking about it, then I will have this session. Okay,
Okay,
because
sir.
in the regular session I only see 2 or 3 people are joining.
Yes,
So
yes.
anyway, I will upload that, record that session also, and then I will upload on my channel. Do like the channel, like subscribe the channel for the regular video update, and follow this page. All the sessions are being shared by the NPTEL team also. You can see that by clicking on the problem solving sessions. All the links are provided here, or you can also subscribe to my channel. You can follow, you can find all the PPT that I am using the sessions in this drive folder. All the all the sessions, all the slides I am uploading to this drive folder, so you can check it there. And also when you are submitting your when you are submitting your feedback, please regularly submit your feedback. And in the separate section, separate question where you have been asked, if if you want to give another comment, you can write a statement like whether this problem solving sessions are important, helpful for you to answer, like the sessions answer while the assignment. So you can also put your comments there. So thank you so much for joining the session. I hope this was helpful for you. And also you can give me your feedbacks related to the session. How can I improve? And thank you so much for joining.
Thank you,
The
sir.
meet. I will end the meeting now. Thank you so much.
Thank you. Thank you.